What an expectant hush has descended. You're all very well behaved. Well, good evening, everyone, and welcome to our Edinburgh Neuroscience Christmas lecture. My name is Malcolm MacLeod, and with Cathy Abbott in the front here, who you'll be hearing a little from later, we're the co-directors of Edinburgh Neuroscience. So this Christmas lecture series has been running for almost 20 years now. And we've had speakers on everything from robotics to neurodevelopmental disorders, from how speaking multiple languages keeps our brain sharp, to how being born too soon can affect brain development. And this year, for the first time, we're particularly pleased to welcome you to the Edinburgh Futures Institute, which used to be, as many of you will know, the Royal Infirmary of Edinburgh. And the motto of the old Royal, which has been adopted as the motto of the EFI also, is carved on the clock tower at the front, and it's Patet Omnibus, which I'm told means open for all, and that's very much our ambition in Edinburgh Neuroscience also. Our research happens because you, and largely the taxes that you pay, which funds what it is that we do, and the pennies that you put in collecting boxes that goes to the charities that then fund the work that we do. And the reason we host these public lectures is in part at least to share the research that we do with you, to hear your thoughts and your questions about it. And so it's a pleasure to welcome you all here tonight. Tonight's lecture celebrates a team that truly embodies, we think, the way that neuroscience research at Edinburgh crosses the boundaries between clinical and laboratory research and looks for ways to work together across disease boundaries to gain really unique insights across different neurological conditions. So Paul, Claire and Sam, who you're going to hear from tonight, established the Edinburgh Living Human Brain Group in 2017, aspiring to develop improved models of human disease. Their group has grown from strength to strength, and that's recently been recognised in an award of £3.5 million from the Medical Research Council to really scale up this work. Of course, using human tissue for research wouldn't be possible without the human patients who generously give the tissue that it can be used. And so that's another reason for our public lecture, to share the insights already gained and plans from the future, as a way in which Edinburgh Neuroscience and the Edinburgh Living Human Brain Group can recognise and acknowledge that incredible contribution of our brain tissue donors to our research, and to thank them for that. So tonight's speakers, we've got Paul Brennan, who's Professor of Clinical and Experimental Neurosurgery and Director of Neurosurgery for NHS Lothian. His research spans the laboratory and the clinic, applying a range of research strategies to improving diagnosis and treatment and outcomes for patients with brain tumours, traumatic brain injury and spinal cord injury. And he collaborates in bringing innovative technologies to the clinic, developing the clinical and scientific evidence for their adoption into routine clinical care. Claire Durrant is an emerging leader at the UK Dementia Research Institute, based in our Institute for Cardiovascular and Neuroscience Research, and her group has funding from many sources, including Race Against Dementia, the James Dyson Foundation, Alzheimer's Society, Medical Research Scotland and the Wellcome Trust. Her diverse team focuses on understanding the causes and consequences of synapse loss. Those are the little bits that nerve cells use to communicate between each other, in neurodegenerative diseases, including Alzheimer's disease, again using a variety of techniques, and most relevant to this evening, including live human brain slice cultures. And finally, Sam Booker is Senior Research Fellow in the Simmons Institute for the Developing Brain, focusing on brain development and how perturbations such as changes to genes or changes to activity lead to alterations in how information is processed. To achieve this, his group examines brain circuits that process incoming sensory information, and they're interested in how those processes go wrong in conditions like Fragile X syndrome. And his group combines the use of living human slices and cultures of human brains in which target genes have been altered to try and understand better the mechanism of these conditions and how we might develop treatments for them. Their lecture will be delivered one after the other, last about for 45 minutes, and we hope that it will inspire many questions, and we've got time set aside for questions after this, followed by, you'll be pleased to hear, a drinks reception. So Paul, thank you very much everyone, and over to you.
So thank you very much for the introduction. I can cut out a bit of what we were going to say now we've had such a nice introduction from Professor MacLeod. As you've heard, I'm a neurosurgeon, and I've been working with and Sam and Claire for a number of years. And in the next 45 minutes, we really want to share with you the work we've been doing, which I hope by the end of the talk you'll see is innovative, it's cutting edge, to try and understand the human brain in a different way to perhaps the way that we've understood it before, and therefore understand human diseases in the brain in a different way. And ultimately, our goal is to try and contribute towards new treatments for neurological conditions, which affect so many of people we know, and maybe some of the people who are here tonight. So we're just 3 people. And we're part of a huge team of other scientists, of research nurses, or the other staff who work in the scientific laboratories, and all the other clinicians I work with. And then there's all the funders, the patrons, the philanthropists who give the money that make this happen. And then, of course, the patients and their relatives who kindly donate the tissue. So it's a big team, and we're just representatives of that tonight. But what brings Sam, Claire, and I together is the focus that if we examine live human brain slice cultures, that we will better understand disease. What do we mean by live? Well, if we can remove tissue from the human brain and we can keep it in that same functioning state, where we can interrogate and observe the cells and the processes and the connections in the same way that they're going on in the human brain, but outside the human brain, that's what we mean by live. And you might wonder how that happens, and we're going to explain how that happens, and then Sam and Claire are going to give you some examples, and then we'll talk a bit about what our ambitions for the future are. But you might wonder why we need to focus at all on the human brain. We've done perfectly well, haven't we, over the last many decades by modelling human disease in animals. We can take Drosophila and zebrafish and rodents and large animals, and we can interrogate the pathways. We can manipulate genes and proteins and molecules in order to probe how disease processes work and how the brain works. And we've generated an enormous amount of knowledge. Why do we need to worry about looking directly at human diseases? And it's true, we have a lot of knowledge. But what we haven't so far achieved to the same degree is that transformative step where we take that knowledge and we actually understand better how to treat people with effective treatments that manage all these neurological conditions. There's a gap, and it's that gap we want to fill. It's not just that the human brain is bigger than some of these other brains or that its folds look slightly different. And although some of the pathways are conserved between other animals and ourselves, there's things that are different, and that's some of what you're going to hear from Claire and Sam tonight. If we can understand those differences, then hopefully we can move ourselves to understanding human diseases better. And I think this quote, for me, encapsulates the problem. This was from 80 years ago. The best material model for a cat is another, and preferably the same cat. So why not for the humans? If we're interested in human diseases, why not focus on live human brain tissue? Well, if you ask that question of a neuroscientist, you'll probably get a very straightforward answer to start with. It's not that easy to get hold of. But there's a secret I want to share with you. It's actually very easy to get hold of if you're a neurosurgeon. And so an important part of what I do in my practice is operating on people with brain tumours. I mean, in the lab, I research brain tumours and how to either stop them growing or just to understand how they grow in the first place. But in the operating theatre, it's my job to take out as much brain tumour as I can, to get tissue for diagnosis, to reduce the pressure on people's brains to hopefully improve their symptoms and so that they can tolerate other treatments. In that process, I need to remove brain as well, and I'll explain why using these 2 scans. So they're both the same scan. I just need 2 scans to illustrate the point. This is an MRI scan, as though I've sliced through the top of your head and just taken the top off. And at the top of the scan is where your face would be, where your eyes would be, and then at the back at the bottom is the back of your head. What's abnormal here is what I've outlined in green. This is a tumour. It's actually a type of tumour called a glioblastoma. It infiltrates in the brain. I need to get in there and get some of the tissue and take some of that tissue out. On the right is what my surgical plan is. And there's a couple of things to highlight here. I can't get all that tumour out. It grows into places that I can't safely get to without harming a patient. And actually, even if I could get to some of those places, there's always microscopic bits of cells I can't get to, and that's one of the problems we're trying to address when we understand these tumours. But if you look up at the top corner at the sort of 1 o'clock position, I need to get into that tumour. I need to get down to it in order to take some of it out. And that processing goes through what maybe looks a bit like normal brain. It's not normal. It's right next to a brain tumour, but it's normally enough that if we can get hold of that tissue, we can interrogate it and understand about how the brain works, how the human brain works, and also start understanding disease processes. So I'm going to tell you just a bit more about that process of how we get tissue. I had wanted to do a live neurosurgery demonstration tonight, but there were far too many forms to fill in. I've seen Tamana here tonight. These images were drawn by her. She's an incredible illustrator, a medical student here in Edinburgh, and she's often in theatre with us, observing the surgery, and she's drawn these sort of step-by-step approaches. Just to give you an idea of how we get hold of this tissue, this is what we do as part of routine surgery, and what, in patients who consent, that tissue that would otherwise normally get discarded, we can use for research. So once we've opened the skin, we're looking at the skull. On the sort of far left picture, you can see 3 little clips down at the sort of 7 o'clock position. That we just put those on the skin to stop it bleeding. And we're looking at the skull. We then have to make a hole in the bone. We just use a drill to do that. And the next thing we see in the third picture along is this big membrane that envelops the whole surface of the brain called the dura, and we incise that. And then it's the first time I'm actually looking at the cortex of the brain. And to be honest, it usually looks entirely normal, just with my naked eye. Okay, the tumour is deep within the brain. There is no clever way I can get from the outside looking in all the way down to the tumour. I actually just need to go through the brain. And so the way we do that is we know a little bit about neuroanatomy. We know where the safe places are to go. We have image guidance systems so that we can direct ourselves to the safest part of the cortex through to the tumour. And then I have to incise the brain. Again, there's not a clever way of doing this. The hole might only be 1.5 centimetres wide, but I need a space that I can get through the cortex of the brain down towards the tumour. So I incise that tissue, I make my cortical window, and then I'm off. I can get on with my tumour resection, usually under a microscope at that stage. So this is what we end up with, and I apologise if anyone's still eating their sandwiches. We get what looks like really nice cortex. As I say, it's been next to a tumour. It can't be entirely normal, but it has that sort of grey-white matter structure that we'd expect from the textbooks. And that's what Claire and Sam will be talking to you about. As we go down deeper into the white matter, we get that junction where the tumour is invading into the brain. And that's a really useful thing for us to investigate when we're trying to understand how tumours invade in the brain, and I'll touch on that later. And then we get the tumour. So these are all bits of tissue that we would usually have discarded, but by focusing on how we can look after them and treat them, we can use them for research. So what we do is, as I'm taking the tissue out, Sam, Claire, or anyone else in their team are stood in theatre waiting. We want to reduce the time between leaving the patient's brain to get into the lab. Because remember, live is about functioning cells. It's not just about cells in a dish or cells on a slide. These want to be alive. So we put them in a liquid, which is an artificial form of brain fluid, cerebrospinal fluid. It's got extra oxygen to keep the cells functioning, and then off to the lab. We'll be there within 20 minutes of actually coming out of the patient's brain. And we can make these ultra-thin slices, so 300 micron thick, 4 or 5 times the thickness of a human hair. And in those are cells which are doing things that cells do in our brains, and we can interrogate them, we can analyze them, often over several weeks. And that's pretty amazing. That's what we've been developing here in Edinburgh. And I'm going to pass over to Sam now, who's going to tell you a bit about what we do.
Right. I'm hoping... Whoops, sorry. There we go. Awesome. I'm assuming everyone can hear me at the back. Excellent. So, hi there. For those of you who don't know me, I'm Sam Booker. I'm a senior research fellow, as Malcolm very kindly introduced. And I'm going to talk to you today about me and my team's efforts to try and really understand how we can use the human brain to develop and improve our understanding of neurodevelopmental conditions. So, as a researcher, I've been at Edinburgh for a little bit over 10 years now, and I've been primarily working within the Simons Initiative for the Developing Brain and the Patrick Wild Centre. And at the core of our research is the mission to improve the lives of people living with neurodevelopmental conditions. And these are often conditions that affect children early in life, either through environmental or genetic causes, that can have devastating effects for their lifelong, their quality of life throughout their early life and into adulthood. And in our centres, what we're trying to do is bridge the lab where I work and feel most comfortable, the clinic where colleagues like Paul and others work, and the community of people living in Scotland with neurodevelopmental conditions to better understand these conditions and improve the treatment prognosis for those individuals. So the work I'm going to talk to you about today has been largely led by a PhD student in the Simons Initiative of Developing Brain who works with us. And that's Max, who's shown up here, if I can work out how to do that. There we go, there's Max. And this is the rest of our team last year at our Christmas night out, so it's topically themed. And what I'm going to try and describe to today is our ongoing research where we're trying to, it's meant to be blank, where we're trying to understand the function of those neurons that Paul talked about and how that relates to the human condition. And so I'd like to start with a simple analogy of thinking about neurons in the brain that transmit electricity around circuits, a bit like the wiring of your house. If you think about the wires in your house, you have uncontrolled electricity flowing around, and without control of that electricity, this can lead to disastrous consequences. Again, topically. The same exists in the brain, where you have groups of cells called neurons that interact with each other, connect, and form these connections that transmit information about the world we experience to each other and around the brain. And when these connections go wrong, this also has disastrous consequences. And we know from lots of research from far cleverer people than I that these processes can go wrong in neurodevelopmental conditions, leading to the plethora of experiences that the individuals have. And so the neurodevelopmental condition I'm going to talk to you about today is called Fragile X syndrome. It affects about 1 in 6,000 individuals. So in Scotland, that equates to roughly about 1,000 people in Scotland. And it results from loss, of this gene called FMR1. And that's found on the X chromosome. And when you lose this gene, and you have alterations in this gene, it leads to this fragile region of the chromosome, as indicated here. And this gives rise to the name Fragile X syndrome. And it's associated with intellectual disability, sensory processing difficulties, so that's how the brain integrates information from the rest of the world and processes it. A number of patients experience autism as well as epilepsy. And so by trying to understand this condition better, we can hopefully improve the therapeutic output and the quality of life for individuals. From a research perspective, we know quite a bit, actually, from these preclinical models that Paul introduced. So we know that loss of the FMR1 gene leads to loss of the protein known as FMRP on the X chromosome, that's coded on the X chromosome. We know that loss of FMRP leads to an imbalance of other genes and proteins. And when we lose these other genes and proteins, we have alteration in brain communication that prevents this passage of sensory information from the world into the brain. So what we're trying to do, we know that from individuals that the processing of that information goes wrong in the cortex that Paul discussed. And we can measure this electrically using electroencephalogram recordings, and we can then listen into the brain and look at the brainwave activity that occurs in the cortex of patients. And when people have done this and looked at typically developing individuals versus Fragile X individuals, you can see using a kind of hot-cold color scheme that typically developing individuals, in response to sensory information, in this case sound, have a response in the brain somewhere around about kind of where the ears are, sort of. And in Fragile X individuals, that activity is higher, and it spreads higher at a higher level around the rest of the brain. So we know there's a change in the electrical activity of circuits of the brain. And we know from other research that rodent or preclinical models, for example, fish, rodents, primates, and also human preclinical models, all share some homology to each other. So they all have neurons, they all sort of look complex, and they all have broadly similar underlying activity patterns. But we're also beginning to understand through the work of Paul and Claire and others around the world that these human neurons that we're talking about have fundamental differences to these preclinical models. And therefore, really, if we want to understand the condition, we kind of have to go right to the source and really understand how these neurons function when we lose genes like FMR1 and the protein FMRP. So taking a step back to think about how processing information could occur in neurons, it's really helpful, I think, to look at the neurons themselves. So these are cortical neurons. These are neurons in the cortex from a human patient that we collected tissue from one of Paul's surgeries. And these are 4 cells here, and you can see this sort of blob around here. This is what we call the cell body. This is where the cells integrate information that they receive on these really elaborate tree-like structures called arbors or dendrites. And there's they receive information from the world on these dendrites, oops, really? Why is that not clicking forward? There we go. There we go. There's the inputs. And they process this information along their dendrites throughout and to produce an output. And so this process of transformation of electrical activity through a neuron is critical for maintaining the fine control of activity around the brain. So what we wanted to do, so unfortunately, one of the main drawbacks of trying to understand Fragile X syndrome in human tissue is that none of the patients that we get brain tissue from, as far as we know, have Fragile X syndrome. And so what we wanted to do was develop a model of Fragile X syndrome using living human brain tissue. So Max in the lab, what he wanted to do to start with was he wanted to look at these neurons that we can visualize, as Paul elegantly discussed about maintained in culture, and to see whether or not we could see FMRP, the Fragile X-related protein, in these cells. So here's a green labeled neuron in living human brain tissue, and Max is labeled FMRP in these cells, and this is shown in purple here. And what I want to highlight is, in this cell, you can see this protein is abundant and highly present inside this neuron, both in its cell body, but also in these kind of dendrites that come out here. So we know these neurons look like what we would hope. They have FMRP. But the real question we want to be able to do to be able to model Fragile X syndrome in this tissue is we want to remove that protein. So how do we do that? How do we remove a protein from a living piece of human brain? We're really good at doing that in rodents and other preclinical models, but human we're not so good at. So how do we do that? So we've got our gene. This is on our chromosome, and we know the gene is FMR1. And we also know that neurons and cells take their genes and read those genes to make protein using a process called translation. And then we get the protein in our cells, in this case FMRP. And we can use a clever trick in the lab using a molecule called a short hairpin RNA, which the details don't really matter for the purpose of this. And this can block this translation process, which means that we can then remove FMRP from our cells. So we can mimic the condition of Fragile X syndrome in some respects. So that's what Max did. So here's an example image of a neuron in green again. And this is where he's introduced this short hairpin RNA. And what you can see is that FMRP is much lower levels in these cells. And it's almost to the point where you can't really see it. And certainly you don't have any structure inside the cell that we saw previously. So we think from a starting point, we have a really good model where we can abolish FMRP and mimic the situation that occurs in Fragile X syndrome. So the next thing we want to do is I find measuring the excitability and electrical activity of cells fascinating. So how can we listen to that electrical activity of a single cell in the human brain? And so Paul really elegantly introduced the idea of the kind of scale of the brain. The scale of neurons in the brain is absolutely tiny. When we think, this is a single neuron here from a human brain, this cell body where we can measure that electrical activity best is about 10 micrometers in diameter. Taking into context, the human hair, so every single hair on your head, if you're lucky enough to have them, is about 50 to 100 micrometers in diameter. So each cell is about 5 to 10 times smaller than a hair. And then to measure that electrical activity, we need to place electrodes onto that cell body. So those electrodes need to be smaller still. So each electrode we use is about 1 micrometer in diameter. So we're handling things under really high-powered microscopes that are about 100 times smaller than a human hair, landing them on things that are about 10 times smaller. And when we do that, we can listen into the electrical activity of those cells. And we can, and this is our cell here, we can measure the inputs. So these are the synapses that process the sensory information around the world, that provide the sensory input. And we can measure the output, otherwise known as spikes. And this input-output transformation is critical to our understanding of how neurons process information. So what Max wanted to do was measure that transformation of information in these cells. So here's an example cell. This has FMRP, so it's our control. And in response to an output, so this is a positive piece of information, this neuron produces spikes. And these are shown here, here, and here. So then Max wanted to look and see what happens in these FMRP lacking cells. And this is shown here. And it doesn't take an expert electrophysiologist to notice that this cell has more spikes, many more of them, than our control cell here. And this is in response to the same input, which is analogous to what happens in the brain, where the same sensory information in individuals with Fragile X syndrome leads to an excessive response of those neurons. In our human model here, we're seeing the same inputs producing an excessive output. So we think this is a really great model to mimic Fragile X syndrome in the human brain. So that's great. So we've got our model. It seems to do what we think it would do. And so then we wanted to kind of really test this system and see whether or not we can provide more evidence that it mimics the Fragile X-like system. So what we wanted to do is we wanted to use potentially as a therapy to try and see whether or not we could prevent this excessive activity. And so for this, there was a clinical trial ongoing, there is a clinical trial ongoing, sorry, looking at a molecule called phosphodiesterase 4D. And it really doesn't matter for the purpose of this what this does. All we know is that drugs that inhibit this protein and this process improve the lives of individuals with Fragile X syndrome. So if we can give this drug to our human system, if we see a correction, this further validates our findings, but also we start to get an understanding of how this drug is working in our system and how we can improve treatments for individuals. So what Max did was he took this drug and the control. So his vehicle is the neutral, inert version of the drug. It shouldn't do anything. And thankfully, under these conditions, we still see our greater output Fragile X-like cells. And in response to the treatment of this drug for 1 day, what Max found was that our control cells don't really seem to do that much different. There are some subtle differences. This drug's not entirely inactive. But in our fragile FMRP lacking cells, what we see is there's a very strong reduction in the number of spikes these cells produce, indicating that we've brought these cells back into the kind of control level where you would expect them to be when they had FMRP. So we think this is a really exciting validation of our model, but also a really exciting outlook for potentially the treatment of Fragile X and devising better therapies. So what I hope I've shown you, and I'll let you guys be the judge of that, is that we have the first model of FMRP loss in the adult human brain. That loss of FMRP leads to alterations in neuronal function, and this is allowing us insight into the function and future directions of new therapies. And just in the last minute or two, I want to touch on where we're going to go next. So I've talked to you about FMR1 and FMRP as Fragile X-related genes. This is a diagram from the Simons Foundation Autism Research Initiative of just all of the genes on that X chromosome that have been associated with neurodevelopmental conditions, where red and orange indicate higher effect. And you can see, hopefully, all of these other genes that we really don't have any idea of how they function in the human brain. And so by understanding more genes and how they affect human neurons, we can hopefully get a better understanding of whether there's commonality or differences between gene effects. One thing that fascinates me is how the diversity of humans as ourselves affects the role of these genes. So by assessing the function of FMR1, for example, across a range of different patients who all have different genetic backgrounds, we can get a better idea for the diversity of individuals living with Fragile X syndrome. And finally, we can use this approach to identify and improve new and emerging therapies. With that, I'm going to say thank you very much, and I'm going to pass over to Claire, and I'm happy to take any questions later on. Thank you.
Well, hopefully my microphone is working at the back, and thank you so much, Paul and Sam, for the excellent introduction. So my name's Dr. Claire Durrant, and I specialise in the diseases at the other end of the lifespan. So we're talking old age diseases, including dementia. So I like to think that makes me sort of the ghost of Christmas future of our little trio, if you like. So unfortunately, most of us in this room will be extremely familiar with dementia and the diseases that cause it. And that's because 1 in 3 of us, without action, will develop dementia within our lifetime. But often when I'm talking about it, people get confused between the terms dementia and some of the other terms like Alzheimer's disease or frontotemporal dementia. What's the difference between this? So dementia itself is not a disease. Dementia is an umbrella term for the symptoms that we see with someone with a disease. So dementia can be, for example, memory loss or personality changes. And just like a headache could be a symptom of a migraine, or more concerningly, or a brain tumor, dementia can be a symptom of a number of different diseases. The most common of these is Alzheimer's disease, but you may recognize some other names such as frontotemporal dementia or dementia with Lewy bodies. But the important thing to remember is that the symptoms someone shows with dementia will differ based on who they are as an individual, but also the disease they have that's causing dementia. So many of you will have seen in the media, for example, famous faces living with dementia. So Sir Terry Pratchett, for example, he had a rare form of dementia affecting his visual system and the way he saw his world. More recently, Sir Bruce Willis has a type of dementia that affects his ability to speak. And I, like probably most of you in this room, have personal experience with dementia. Now, this is my grandmother, Sheila. She died with Alzheimer's disease in 2018. And I always like to show her picture in such talks for two different reasons. First, she had what we'd consider classic Alzheimer's disease, so really a problem of memory loss. And as you can see, she attended my wedding. She had a fantastic day partying with the best of us. But 2 days later, when we visited her, no recollection whatsoever of me having been married. So really losing these things. So what we did is we showed her this picture, and she went, "Ah, of course, what a lovely picture of you and I. But who is that old man standing next to us?" Now, all of us in this room can see that that old man is a bunch of flowers. But for her, she wasn't taking in information about colour, texture, or context. She was almost trying to make the dark patches in that image into a face. And for me, as a neuroscientist, that was the starkest reminder I've seen that Alzheimer's disease can be so much more than just memory loss. The whole perception of your world changes. So I always include this quote from an author called Wendy Mitchell, who lived with dementia and wrote a fantastic book called Somebody I Used to Know. And she says, when you meet one person with dementia, you truly have only met one person with dementia. So my aim as a scientist is to try and understand the diseases that really cause this horrible, horrible symptoms. And the most common of these is Alzheimer's disease. Now, what we all want is a cure. So why don't we yet have a cure? And the sad reality is that it's an extremely complicated disease. When we look inside the brain of someone who's died with Alzheimer's disease, we can see quite clearly there is a massive death of nerve cells. The whole brain is shrinking, and you start to see little holes appearing. And one of the holes that appears is in a region called the hippocampus, responsible for short-term memory and navigating your way through space. Links quite well with the symptoms you'd see in early Alzheimer's. But the way we know for sure this is Alzheimer's and not another type of disease is when we look at a microscopic level, we see two key hallmarks. The first is some clumps of protein that exist outside the cells called a plaque. This is formed of protein called A-beta, or amyloid, you may hear in the media. The second is another set of protein clumps, but this time inside nerve cells. These form structures called tangles that are formed of clumps of protein called tau. Now, we know through decades of experiments in the model systems Sam and Paul was talking about that toxic forms of these proteins that are clumped are deadly toxic to synapses. Now, this is the most important structure in the brain. This is the connections between nerve cells that allow them to communicate with each other. If these structures die, the brain is silent and you're not able to express things. And this is why we see dementia. So to have a cure, we need to protect these structures. Now, in mouse models, we've had quite a lot of success in curing Alzheimer's disease, but unfortunately, this has not really translated to huge breakthroughs in the clinic. Now, it might be, as Sam and Paul mentioned, that we have massive structural differences between a mouse and a human brain. But it could also be quite simply that mice naturally don't develop Alzheimer's disease. In order to study Alzheimer's disease in a mouse, we have to pump them full of artificial, highly mutated versions of A-beta and tau to give them even vaguely the same symptoms we'd see in a human. So what we really need is to be studying these processes in living human brain. And that's what my team have been doing in collaboration with Paul and Sam and so many people sat in this room, is using these pieces of brain to explore early stages of Alzheimer's. Now, most of you listening to Paul will realize that our samples, they don't come from individuals with Alzheimer's disease. They come from individuals with brain tumors. But we noticed that the average age of the samples we were getting was about 65. Now, rather horrifyingly, it takes about 20 or 30 years from the early changes associated with Alzheimer's disease in your brain to actually cause you to have problems with day-to-day life. So it's highly likely that people sitting here in this room will have the very starts of Alzheimer's there, but won't know about it for years later. Is the same true for the samples that we get from the clinic? So the first experiment we did was to simply look for A-beta plaques, and we found them, and tau tangles. And in about 30% of our samples, we see some of these very early changes that gives us a unique opportunity to perform experiments, watch them in real time, and understand how they might interact with living human brain. But not all of our samples have Alzheimer's disease, so we have to do some other clever modeling as well. So when we're using human brain slice cultures, the first thing we wanted to know is, does the cell type that contains synapses survive in culture? And we're looking at neurons, these beautiful structures that Sam showed, and I think they just look absolutely stunning. I'm a nerd, I love pictures like this. Beautiful sort of trees-like structures in the forest, and we know they survive for weeks in culture in our hands. But even better, they're sitting at our mercy, and we can perform experiments without worrying about doing any harm. And what we can do is do some rather horrible things to living pieces of brain. We can throw in toxic proteins, and by toxic, I mean that A-beta and tau that we see in Alzheimer's, and watch what happens. We can throw in experimental drugs and see if we can interrupt that process. And crucially, we can actually understand what happens. So we can take bits of the culture medium, that's the liquid that's keeping these pieces of brain alive, and look for changes in proteins. And we can also use really great microscopes to actually watch at the level of the synapse how things are changing in early Alzheimer's disease. So with the rest of my time, I'm going to give you two little experiments that we've done to give you an insight into what we do in the lab. So one of my first questions I wanted to ask is a big one in Alzheimer's disease. Why do different parts of the brain seem to suffer more strongly with disease than others? And if we look in Alzheimer's disease, the first area to really degenerate is the temporal lobe. Now, you may realize that the temporal lobe is so named because it's on this side of your head, and it's named because it's the first part of your hair to go grey. So it tells your age. It's the temporal lobe. It's also the first part to be affected by Alzheimer's disease. Now, the frontal lobe is affected a bit later, and when we look post-mortem at people who've died with Alzheimer's disease, we'll find far more of these big structures of tau in the temporal lobe than we will in the frontal. But what is it about these regions that mean they're more likely to form these clumps of tau? So we did a really simple experiment. When Paul is taking brain tumor tissue, he only takes it from the region where the brain tumor has popped up. So we get a nice mix of different brain regions arriving in the lab. So we thought we would just see in different regions, do we see different levels of tau being produced by brain slices? Bearing in mind, these are from individuals as young as 28, so with no expectation of Alzheimer's disease. And what we find astonishingly is in the temporal lobe, so this area that's highly vulnerable to Alzheimer's, we see almost double the level of tau being released out of these cells at baseline. That happens over your lifetime. That might explain why this region is being hit first. Next, I really wanted to understand why and how do synapses die in response to these toxic proteins. So we switched ways a little bit. We decided to focus on A-beta. So there's a lot of evidence from mouse models and cell models that toxic A-beta binds to and destroys synapses. But we've never seen this in living human brain. So we did an experiment where we took brains that had been donated by individuals who died with Alzheimer's disease, who wanted their brains to be used for research. We extracted the toxic A-beta from these brains, and we added it to our human brain slices. We then looked at a really tiny level, so I'm going to go even lower than Sam. We've got less than 1 micrometer here, so we're talking 1/100th of a human hair at what happened at the synapse. And what we see here is the synapse is labeled in yellow. This is the communication point between the nerve cells. Is the A-beta labeled in pink is hugging onto this synapse. It grips and it holds onto it for dear life. And what happens when it holds onto that synapse is when we treat with A-beta, we see a loss of synapses. So we go from a normal level of synapses to a lower level. So we've seen for the first time in living adult human brain definite proof that A-beta can kill synapses. So I hope in my very short tour of what my lab is trying to work on that I've convinced you, or at least that I am convinced, that the future of Alzheimer's disease is in human. I want to know why these brain regions are vulnerable. Can we stop it at the source and try and interrupt that? We've got this amazing ability to kill synapses in a dish. Can we interrupt that with new drugs and test new therapies? We've also got a natural model of disease. Whilst we don't know for sure that the A-beta plaques and the tau tangles we see in our samples would become Alzheimer's disease, we can do lots of experiments to understand how that changes real human brain. And I hope you think, like me, that if we can take the best-in-class treatments developed by amazing work in other types of models, like mouse and slicers, that if we test them in the human slices before going into clinic, if it works in living human brain, hopefully it's more likely to work in living humans. And finally, this is sort of my indoctrination. My view is that we really need to fight against this. We need to accelerate research to a world free from dementia. If 1 in 3 of us will develop dementia in our lifetime, I hope that my sons, who are 4 in 1, have better odds than that, and they can have many Christmases free from the heartbreak of dementia. So I'm just going to pass back to Paul, who's going to give us a little bit of brain cancer insight, and then on to the future, but really warmly accept any questions after my talk. Thank you.
Great, thanks very much, Claire. So you've heard how we can use these amazing live human brain slices to understand neurological diseases. And the tissues come from patients with brain cancer, so it's really important that we're researching brain cancer as well. So that's where my work is. We don't have time to go into the brain cancer detail. I just want to pick out one really exciting thing where I think the live human brain slices are the only way we can really try and understand a particular type of problem which is really relevant to human brain disease. So for a very long time in many cancers, we think about the tumor, the cancer, as the tumor cell. That's the bad thing. If only we can get rid of that cell, we're totally sorted. And we spent many years in cancers like glioblastoma and glioma trying to extract those tumor cells from the tumors and to understand their genetics, the way they live, and to try and kill them. And it just hasn't worked. We haven't fundamentally changed the treatment for glioblastoma in 20 years. And we're now much more aware that these cells don't exist in isolation. They interact with all the other normal cells in the brain, so the neurons, the blood vessels, the immune cells which are in the brain. And there's this constant trade-off between our body trying to attack the tumor to stop it growing, and possibly even the tumor cells trying to hijack the normal mechanisms in the brain. And that microenvironment, we can't model that by chucking loads of different cells in a dish. We can't really model it by trying to use a rodent model where we're trying to put a human tumor into a rodent brain. We need to use human brain slices. So I just want to give you a quick insight into that and about how that can really transform our understanding about human disease. And Alzadeg, who's here, is an excellent PhD student who's done this work, building on some really great work by Shahida. I don't know if she's here tonight, who I've worked with for a long time, and amazing images produced by Justina on the microscope. It's a real team effort to produce these things. What we've got on the left is this same brain slices that we've taken out of theatre. So as we said, they're on the edge of where the brain tumour is. The tumour is not yet invaded into that part of the brain. If we could stop brain tumours invading, actually we could go a long way to helping treatments like surgery and radiotherapy being more effective. So we can take human brain tumour cells from other patients labelled in green, and we can put them onto these brain slices. And here we've stained the brain slice with something that lights up an immune cell called a myeloid cell, which has got a role of sort of navigating around the brain, trying to mop up things that shouldn't be in there. We want you to just focus on what the green cells are doing. This just lasts a few seconds. This is more than 30 hours worth of observing these cells. So as those tumour cells, which we've just dropped or placed delicately onto the human brain slice, you can see them invading into the brain. Okay, and you can see the proximity of those invading brain cells to the myeloid cells. So we can start asking now, what is the role of that interaction? And rather than treating the tumour cells, is the way to address the tumour cell invasion actually to somehow manipulate those myeloid cells, to almost downgrade and turn off the tumour cells? It's a totally different way of thinking about how we treat tumours or how we understand tumours, and we wouldn't get that unless we use these live human brain slices. The second image just looks at this in more detail. I just want to dive into that interface between the tumour cells and the myeloid cells a bit more, because it's not as simple as one or the other. It's not do the tumour cells get eaten by the myeloid cells, or do the myeloid cells support the tumour invasion? Focus first of all on these two yellow arrows. So we've got a myeloid cell in red and a tumour cell in green. So what happens first of all? This myeloid cell is eating the tumour cell. Okay, this is a real human myeloid cell, a real human tumour cell in a real bit of human brain. And this is what happens. The myeloid cell has the job of trying to get rid of tumour cells or infection or whatever it is, and it's doing exactly that job. Imagine if we could persuade all the myeloid cells in a tumour to do this. More than 50% of the cells in a human brain tumour are myeloid cells. We would have totally nailed a cure for cancer. But it can't be that easy. If you take a look at the white ones now, this is another myeloid cell and another tumour cell right next to each other. But instead of getting eaten, there's some sort of interaction going on. Is this where the tumour cells somehow hijacking the myeloid machinery in order to maybe invade further into the brain, or maybe in terms to survive? So these are complicated multicellular interactions in a complicated multicellular environment. And we can only really start to understand these if we use live human brain disease. So I think this is a very exciting time for us to be doing research, and it's great to be able to work with Sam and Claire and everyone else in order to achieve that. And what's next? You heard from Professor McLeod that we were fortunate enough to get a big cash injection from the MRC welcoming UKRI. How can we do better than what we do? Well, we're lucky enough that Edinburgh is now going to be the national centre for live human brain slices, and we're going to have a role in training other neuroscientists around the country to do these slice cultures well, to be able to get the slices to last for as long as possible. And the hardest bit, to collaborate with neurosurgeons to get hold of the tissue. Okay? But we want to know more about the patients. We want to understand more about their life journey, about the diseases they've had, to assay things in their blood and elsewhere in the body to understand their disease process. So, as you heard from both Sam and Claire, we can model individual diseases. But imagine if we could model lots of different diseases. If we understood a lot more about these patients before they go to surgery, we can work with other researchers on different diseases, and we can really make sure that the human disease is modelled in human beings. It's got to be the best model of human disease. And so I hope over the next few years we'll be able to come back and tell people about the big transformational steps. But what we really wanted to do is support all neuroscientists to make this part of their armoury in order to get to there. So just to recognise that it's not just us three by any means. There's a huge number of people involved in making this happen, and ultimately maybe some of the people here have donated their tissue to us. We wouldn't be able to do it without your help. So thank you very much for listening. Merry Christmas, and happy to take any questions. Thank you.
Thank you so much. That was absolutely amazing. So we'll take questions for the next 10 minutes or so. If you have a question for somebody specific, then by all means say so. Otherwise, we'll just direct the questions to whoever seems more suitable. And Maria's coming round with a microphone now.
Thank you. As somebody who volunteers for the biobank, and my brain has been scanned a few times, I've done lots of memory tests and cognition and speed of reaction tests. I'm sure my genetic code has been analysed. Do you interact with what the biobank is doing, actually?
Yes. I thought you were about to donate some brain. I was going to say, we've got a little facility outside if anyone does want to. So, yeah, I mean, what's interesting is a lot of people go through the biobank do inevitably end up getting surgery. And it's the same in neurosurgery as it is in other surgeries. So we interact in that way. And then we have a live course of history. But also the biobank gives us a big denominator. We're able to interrogate those data sources to understand more about the spread of whatever feature it is or disease process in the population that we then map onto our patients. So we can only ever get tissue from a relatively small number of patients. If we know more about communities, and the biobank is a good way of doing that, we can understand much more from that population about the small sample of tissue we have.
Question over there.
Hi, I hope you can hear me okay. So my question's about medicine. Can you hear, is that better?
Yeah.
Yes. So I'm a nurse and I work in primary care, but my question's on advancements of human interaction with technology of the future. And the question, I had to write it down, so bear with me. So in human neural networks, meaning and behaviour emerge from relational patterns rather than static rules. So think about AI systems also being trained through relational patterning. Do you foresee a point in the future where similarity in learning dynamics requires a new ethical framework for human and AI interaction? And if relational architectures, biological or digital, form patterns that evolve, adapt and respond to AI relational awareness, how should we ethically recognise the unstable boundary between tool and relational partner? And do you think that medicine is ready for that?
I think that's quite a big question. Probably not one that any of us can address right here, but happy to chat with you afterwards about that.
Sure,
Yeah.
that'd be great. Thank you.
Yes. Over here.
The brain has got a very big population of cells, and you've taken out a tiny section and put them into a concentration camp. Are they going to behave the same way as they were behaving before?
That's a really good question. So obviously, we're never going to capture networks that span the whole brain. So there's lots of neurons that send axons all the way over to the other side of the brain. What we can capture, though, is that your brain is, if you imagine an ice core in the Arctic, when you sort of go down in a borehole and the sort of little strips that we get, we have a unique architecture full of layers. And we capture that in every single slice. So there's 6 layers, and they all talk to each other in these things called microcircuits. So we can really look at these small interactions across the whole layers of the brain. And we're aware that we're missing, obviously, big aspects, but we know that a lot of the damage that happens on the microcircuit level will affect the whole circuit. So we can extrapolate to some degree in that way.
And this one up there. This microphone can be thrown, but you need to be a rugby player to do it. So.
That was really great, thank you. This is a question probably for Dr. Brennan. I'm curious what characteristics you're using to determine what areas of the neocortex you want to go through that you referred to as being safe to go through.
Yeah, safety is relative, isn't it? I mean, the main requirement is it has to be near to the tumour I'm trying to get to. So when we're making decisions about doing tumour surgery, there's some people we don't offer surgery to, or we don't offer the same surgery to because we don't think we can safely get there. What's amazing is quite how resilient the brain is. There's lots of areas that on a map of function you think you can't get very much further into the brain than to stare at the outside, but it's amazing what you can take out. So we're building on experience of lots of people when we've done things before and the patient's been okay afterwards. So I think it's anatomy more than function. But I guess you raise a point, which is if we knew more about the function of these different bits of the brain, would we change our approaches? And I think these are sort of aspects that it's probably going to run along side by side. But yeah, anatomy is the best friend, I think.
I will add to that, as one of the scientists on the receiving end of the tissue, we have many, many cases where we turn up and wait for 3 hours and Paul says it's not safe to give you tissue. That happens all the time. So it's very much that the only tissue Paul's taking is tissue he would take if we didn't exist. This is tissue that would otherwise go to waste. So we're very, very happy to spend hours waiting on the off chance that there's a piece of tissue that's safe to take for us, but we're very happy on the days that we don't. It's just the nature of working with human tissue.
Hi, I had a question about ethics for you, and what I was wondering is, in working with these brain slices, you are potentially going to be able to identify diseases like Fragile X premutation or early symptoms of Alzheimer's. And I was wondering, in those cases, do you inform the patients of this, or is it left as their decision, or basically what's the ethical?
It's a tricky question because ultimately I think one of the main considerations is that a lot of the approaches we take in the lab would be considered experimental biology anyway. So determining a diagnosis or a indication or a sure diagnosis would be very ropey based on our approaches. However, we have had these discussions about if you were to find a patient that was had particular evidence of, say, neurodegenerative pathways, what is the discussion point to have? I don't necessarily know if we've come up with a good ethical standpoint for that. But
I mean, the ethical framework of when we consent patients, we actually make them aware that we wouldn't tell them. Because as Sam said, unless you actually know for certain that, or the same with Claire, unless we know that identifying a plaque means that you're going to get Alzheimer's, we've not really added any value to your life. And since we don't have any treatment for Alzheimer's disease, we wouldn't. So, but these are all real issues. And particularly when we start developing the centre, I think having a forum where we can sort of discuss these things, because as the treatments do become better and as the tests do become better, these are real problems that we might, so I think it is relevant for us to think about it.
Another question over here.
Yes, so I wanted to ask, so you're modeling your things on essentially tumor-adjacent tissues. So how are you sure that the changes you see are in response to your manipulation and not some predisposition that the tissue might have had due to the tumor?
So the simple answer is we can never be truly sure that what we see in these samples is going to be representative of the entire population. What we do know is that our colleagues who work on different types of tissues, so for example epilepsy or hydrocephalus, which are other sources, don't really see big differences compared to what we see. We also, when we get tissue, we usually get enough to divide it into lots of different dishes. So for every patient, we're not just doing one treatment. We can do a control to see what it looks like normally, add a toxic protein, does it get worse, does it get better? So we're always comparing to what's normal for that sample, not what's normal for the population, which help us to interpret a few more things.
Any last yes, one more question here.
Hello, thanks for a great talk. You talked about the genetic backgrounds and so on. So I was wondering, how often does it happen that the effects don't generalize, that you see an effect in one brain slice that you don't see in another one? And so your thoughts?
I guess in some ways that's the benefit of what we're trying to do in the future. Imagine if we could work out the genetics before we took people to theatre, so we could actually stratify patients based on risk for disorders which they might not have, but they have risk for. So I don't know, Sam, if you want to talk about.
I'll give a slightly different example. So actually, one of the considerations we have is, these patients all have diverse clinical phenotypes already. And one of the big clinical phenotypes in glioblastoma is that approximately half the patients will have experienced seizures and half of them will have not. And even at that level, just that level of granularity of has the patient had a seizure or not, even though this is not the seizure focus, this affects the function, the underlying function, and somewhat the structure of the cells in the brain. So even at that level, we still haven't, as a neuroscience community, haven't worked out what is the important information yet, let alone what's happening with diversity of genes.
Right, one last question over here.
We know that the brain can be affected by a number of external factors, such as concussions, plastics potentially, fast foods, ultra-processed foods. Is there potential to study those things with these cells as well?
So one of the things we're introducing will be surveys on lifestyle factors. We already take things like BMI. Just general data about lifestyle would be really interesting as well. We can also do things, so we have colleagues working at other institutions who do things like adding pollution particles to other cell types. We can collaborate with them and look at the effect on the brain. So certainly, where there's a molecule that we can add that might mimic some of these changes, we can do that really easily. And where we can get information from the patients about what that brain has experienced before it ends up in our lab, that's what we're hoping to do.
All right, one last one over here. Sorry. Here, yeah.
What you've described is a huge research undertaking that's going to take many, many years to achieve its full potential. Is it a real challenge to put together the funding to sustain this over such a long period?
It was until 4 weeks ago. It's been quite, these questions that people are asking are very good questions. You could easily go away with the view that it's not reproducible, it's difficult to get tissue, you're not really looking at the whole brain. There's lots of reasons you can come up with why this isn't a good strategy, but I think where we've gone from when we started to where we are now, the work that you've heard that Sam and Claire have done, it's really groundbreaking. And there's lots of other examples of that type of work. So I think we moved to a position where people do accept that this is a useful model. We're not saying that other models are no good. We're saying that this is a model that you should add to your armory. So it does feel as though that's getting some traction, and hopefully, you know, we can be more successful. But the bigger limiting factor is there's only so many people who get brain tumours. And so what we really need is we need neuroscientists all over the country, all over the world to be doing exactly the same thing. And then we'll get a critical mass. So I think that's, for us, the usual question is, what happens when you run out of tissue? And we haven't yet, but we will do at some point. And so, you know, we're building a network across Scotland, across the UK. And that network will only work if people buy into the idea that this is worthwhile. So hopefully these sorts of lectures convince people about that. Yeah.
Thank you. So the word inspiring gets bandied about a lot, but this genuinely was. Those were amazing talks, fantastic questions from all of you in the audience. Thank you so much to the speakers. Thank you to all of you for coming out on this cold night. Particular thanks to Maria and Deborah, who have organised the whole thing and done an incredible job, because there's a huge amount of attention to detail goes on with these. There'll be a feedback form coming round. We'd love to hear back from you, ideas for future years, what worked and what didn't. Thank you all very much.
Thank you so much.